Muhammad Umar Farooq Biological Video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also question answer to each other for solving the problem. So today we will discuss about the synthesis of cholesterol. So the cholesterol is the diagram is like here, that is the four carbon containing rings and one chain, smaller chain, and is the cholesterol is the important for production of hormone, steroid and plasma membrane. For the plasma membrane, you should need to know about that the plasma membrane is sportive property by the water soluble activity, and that spores which that is gap is created during the plasma membrane phospholipids bilayer, which that create a gap and that's fill the gap for uh, further processes. So here I am drawing the lever, which that is we should understand the cholesterol for transport in our body. So we will discuss about the biochemistry in this video, but here is the introduction. You should understand the overview of the cholesterol. So here is the acetyl-CoA will be produced from the glucose during the glycolysis to go pyruvate, and that pyruvate go into the mitochondria, and that will be converted into cholesterol. And this cholesterol, as well as here is the bile acid can be production, and lipoprotein, high density lipoprotein and low density lipoprotein will be produced. So here is a bile duct and contain a gallbladder. That is gallbladder contain a bile acid and bile pigment, which that is the byproduct of the breakdown of red blood cell. So here is the cholesterol, and one more thing is important things is apoprotein and phospholipids and cholesterol and triacylglycerol, which that's combined together to give a this structure is look like this. That is the apoprotein, and it is known as high density lipoprotein because apoprotein is more. So this here is the digestive tract, duodenum, which that is the open bile duct, which that's open and excrete the bile acid and fats and cholesterol, which that is extra in our body, which that is excrete extra bile and extra salts, bile salts, fats, cholesterol is excreted through the intestine. So here is the liver contains scavenger receptor, which that is receive back the excess amount of lipids, excess amount of cholesterol or fats. So here is the different type of tissue which that is absorb the cholesterol and fats, lipids, phospholipids and protein, I mean in the form of amino acid. So this all thing is transported by the uh, high density lipoprotein part, which that is made from liver. So here the tissue will produce hormones, steroids, phospholipids, triglycerides, phospholipids. And here is the adipose tissue, and muscle use amino acids and fat as well as phospholipids for plasma membrane. So the here is the phospholipid bilayer of plasma membrane, which that is contain red color shows that the cholesterol, and this cholesterol is used for the water soluble prevention, where the need and where the gap is left behind, that is the filling mechanism. So here is the liver, which that is absorbed from the bloodstream glucose from the glut channel by the active transport using ATP through two sodium molecule will enter with the one molecule of glucose, and this molecule will go into the glycolysis to produce 2 pyruvate, and this 2 pyruvate will go into the mitochondria to produce acetyl-CoA, and this acetyl-CoA will go into tricarboxylic acid or citric acid cycle or Krebs cycle, or either go out or excrete exit from the into the cytosol, and that's 2-acetyl-CoA will convert by the presence of enzyme thiolase to produce acetyl-CoA, and this acetyl-CoA is the major function to produce by the chemical reaction to produce cholesterol. So this cholesterol is look like this. And on the other hand, the acetyl-CoA one molecule separate from two and that's convert into the malonyl-CoA. In the presence of hydroxymethyl CoA reductase, HMG CoA reductase enzyme to use to produce malonyl, and this malonyl will be converted into the glycerol to monoglycerol by the monoglycerides and diglyceride and triglyceride by one converting of malonyl CoA into a fatty acid, and that fatty acid will be attached to form a triglycerol. On the other hand, here is the hydroxymethyl glutaryl HMG CoA reductase enzyme used. So here is the statin is the medicine drug which that inhibit the chemical reaction of acetyl-CoA to convert malonyl-CoA. So here is the triglycerol will be formed, and apoprotein, phospholipids and cholesterol fatty acid will be formed, and these apoprotein, phospholipids, cholesterol and fatty acid as well as triacylglycerol will be come together and go into the Golgi bodies, and these Golgi bodies transfers to produce a mature high density lipoprotein which that is empty mean which it is able to give the amino acid phospholipids to a different region of the body, flow into the bloodstream, and that's where need they give a complete uh, complete exchange of amino acids, and where they have not need to use the fat in the tissue, they will absorb to become a full high density lipoprotein, and this become a low density lipoprotein which that's go into the liver, and this go into the liver to excrete the high cholesterol through the bile from here. So here the scavenger receptor are present which that is excrete the bile, excrete through bile. It can be a fatty acid, lipoprotein, or it can be a bile acid or cholesterol. There are many varieties which that is excreted from the bile, and that will be reabsorbed 10% from the large intestine and small intestine by the bloodstream, but it's um, we will discuss in the next video about this reabsorption. So here is the synthesis of cholesterol by the mechanism of biochemistry is here. Here we will discuss about the complete information to understand the chemical reaction. So here is the 2-acetyl-CoA which that is used by the thiolase enzyme, and additionally CoA which that is the used to release CoA will be separate, and this one CoA will be formed, and that's acetyl-CoA will be formed, and this acetyl-CoA will be attached by the acetyl-CoA will release their two carbon to produce here is the uh, 3-hydroxy-3-methyl glutaryl CoA. It is known as HMG CoA, and that is the HMG CoA synthase enzyme actually used for it, and acetyl CoA will be converted into a CoA, and that CoA SH, and that two carbon will be transferred to the HMG acetyl CoA, and that's here is a statin is used to prevent the HMG CoA enzyme. Uh, so the here is the drug which that is prevent known as example simvastatin. So to NADPH is used to HMG-CoA reductase enzyme used to convert a product is known as mevalonate. Mevalonate is the very important product. So here the bile acid decrease of the bile acid, cholesterol decreasing, and mevalonate decreasing to stimulate the HMG-CoA reductase to produce mevalonate for making a cholesterol. So that if low cholesterol, low bile acid, low mevalonate, that is the stimulation point which that stimulate to HMG-CoA reductase to produce mevalonate. So the further is the mevalonate kinase enzyme used to ATP using actively mevalonate kinase enzyme by the 2 magnesium ion to produce mevalonate 5 phosphate. It's a mevalonate 5 phosphate means the carbon number 5 contain a, a phosphate group. So it means the phosphate group. So here is the mevalonate 5 phosphate, which that is phospho mevalonate kinase enzyme used to produce it. So the phosphate group will be added by the ATP to convert ADP. So here I am discussing about the mevalonate 5-phosphate, which that is on the active of diphosphomevalonate kinase enzyme to by the using of ATP. Obviously, that's convert into the mevalonate 3-phospho 5 diphosphate. It means the two phosphate group are attached. The carbon number 5. On the other hand, carbon number 3 contain a 1 phosphate group. And here is the carbon dioxide will be released, and one inorganic phosphate is also released to produce a diphosphomevalonate decarboxylase. So the diphosphomevalonate decarboxylase enzyme is used to produce isopentenyl diphosphate. So the isopentenyl diphosphate is the here is the basically carbon number 1 contain a double bond, carbon number 3 to 1. So is the isomerase enzyme used to here is the bond which will be exchanged. So this bond will be exchanged here to here. So here the product will be formed is known as 3,3 dimethyl allyl diphosphate. So the 3,3-dimethyl allyl diphosphate will be formed, and this is the very important character. For example, the isopentenyl phosphate is further used also, and here is the HMG-CoA will be also produced by the transmethyl glutamate shunt. It is known as transmethyl glutamate shunt. We will not go into the details, so just remember about that. It means the 3,3-dimethyl allyl diphosphate will can be produce uh, can produce HMG-CoA. So here is the 3,3 dimethyl allyl diphosphate will be used by the enzyme is known as cis-pentenyl transferase enzyme to produce one isopentenyl diphosphate is used in the presence of cis-prenyl transferase to produce by the releasing of two phosphate group to produce very important organic molecule is known as grenyl diphosphate. So this grenyl diphosphate will be produced, and that further is same reaction is the cis-prenyl transferase enzyme by the activity to further one more molecule will be added. So the carbon 11 contain 11 carbon containing compound. So the 11 carbon containing compound will be formed, and that is known as grenyl diphosphate. And that's again cis-prenyl transferase enzyme used to release the 2-phosphate group from the isopentenyl diphosphate, and that is the same reaction will be occur and produce uh, 5 more carbon addition. So it means the 11 plus 5 add more to produce 16 carbon containing compound is known as farnesyl diphosphate. So this farnesyl diphosphate is can be go into the dolicol, is a cis-prenyl transferase enzyme using to produce dolicol. So here is another compound will be formed, but here we will discuss just cholesterol synthesis. So the this molecule is many property which that's convert into the prenylated protein by the addition of amino acid or uh, or transprenyl transferase enzyme to make a side chain of ubiquinone, which that is the antioxidant of the human body. And on the other hand, heme group A will be found by this compound that is known as farnesyl diphosphate. So the major reaction we will carry here, the squalene synthetase enzyme to produce by the releasing of 2 phosphate group on the action of NADH to produce NADP plus, and that's magnesium and manganese as a cofactor used to produce squalene. That's a squalene is the very important organ which that produce cholesterol. But on the other hand, one more thing is about that. Here is a transprenyl transferase enzyme used to produce side chain of ubiquinone and heme A. Here is the here is the half molecule of oxygen used as well as NADPH used to produce squalene epoxide. So the squalene epoxide is contained like this, and this is the carbon is the rearrangement from here, and that rearrangement is the carbon number carbon number 4, which contain go into attach to become a ring formation. So here is the carbon number 13 attached by the methyl group to form a ring structure, and carbon number 14 is attached. But on the other hand, one reaction is more acetyl-CoA will be converted into the by the chemical reaction to produce mevalonate, and this carbon dioxide and water will be excreted to produce isoprenyl unit. That is, isoprenyl unit is the precursor of sex hormone, steroid, and androgenic hormone, or basically steroid. So this isoprenyl unit, 6 prenyl unit, is the by the chemical reaction to produce choline. Again, this is the separate chemical reaction. So here is the oxidosqualene lanosterol cyclase enzyme used to produce lanosterol. That's a lanosterol is a ring formation, as carbon number 4 contain a ring by the action, and here is the hydroxyl group will be attached, and that's the carbon number 14, which that is again go into the carbon number 13, and that is the chain is also as well. And that's after the lanosterol, that is the carboxylic uh, carboxylic group will be released, and NADPH used as well as more oxygen used to produce 14 dismethyl lanosterol. So the 14 dismethyl lanosterol is the basically uh, carbon number 14 contain a dismethyl. It means the not methyl group attached. So it means the release of the. So here is the 14 dismethyl lanosterol will be formed, and this further go into the by releasing of carbon dioxide and using the oxygen and NADPH to produce a zymosterol, and this zymosterol is the hydroxyl group will be attached, and their structure is like that, and carbon 14 contain a double bond. And that's isomerase enzyme used to produce delta 724 cholestadienol, and that's produced here is the 14 carbon number, and 7 is here, which that contain a double bond. 7 and 24 carbon containing double bond is formed is the character of delta 724 cholestadienol. Di mean to enol, and that is the major important. So the NADPH use and oxygen use again to produce dismosterol, and 24 dihydrocholesterol. That's a 24 carbon number 24 contain a dihydrocholesterol. It means the chain is going down, and there's a 24 to go a dihydrocholesterol. That's a dihydrocholesterol, which that is the precursor of steroid, and that is here is the cholesterol will be formed by the delta 24 reductase enzyme and NADPH used to produce cholesterol. So this all is overview of the cholesterol synthesis and the biochemistry, as well as overview of the cholesterol synthesis and the chemical reaction and their physiology. So thanks for watching.